Hi, welcome everyone. Thank you so much for joining us today. I'm Alicia Newton, and I'm the Director of Science and Communications here at the Geological Society. I'm so pleased to welcome you today to our first virtual public lecture. Back in January, we marked the start of the Year of Life with a whole range of activities planned, ranging from art exhibitions to conferences, all intending to be held at our London location at Burlington House. Obviously, that didn't quite go to plan, but we're pleased to now be bringing as many of these activities online as possible. And I hope you'll take the chance after this talk to go visit our virtual events calendar on the GSL website. So today we're starting a virtual public lecture series with a talk by Professor Daniela Schmidt. She's a professor of paleobiology at Bristol University. And I just want to say that today's lecture is hosted in conjunction with the Linnaean Society, who are our neighbors across the courtyard in London. So Daniela studies ancient marine ecosystems and how they've varied through time. Over the next 40 minutes, she will explore how ecosystems have responded to past changes, such as warming or ocean acidification, and what clues these changes can offer for understanding the future of our oceans. There'll be time for some questions at the end. We won't be able to call on people, so if you do have a question, please type it into the Q&A box. To do that, if you look at the bottom of your screen, there's an icon for Q&A. You just have to type the question in and hit send, and we'll try to get through as many of these questions as we can before the end of our lecture. So please join me in welcoming Daniela. Thank you for being here today.
Thank you very much, Alicia, for having me here. And thank you all for joining in this slightly unconventional way of giving a presentation. When I was asked by the Geological Society and the Linnaean Society if I would give this, I had not expected to do this in my own home. I've done a lot recently via Skype and or other media, and you can see here the way I'm currently interacting with my group. And so I'm hoping that will have trained me to talk to you over the next half hour or so about some of the research we're doing as a group. What I want to show you is first how we detect climate change, what the projected impacts are of climate change on ecosystems, and the lessons we can learn from the geological record. This is a graph many of you are familiar with. And what it shows you here is atmospheric CO2 in ppm versus years. This is the very iconic Mauna Loa record of CO2 rises in the atmosphere. You can see the seasons going up and down due to the changes of photosynthesis drawing down CO2 in the atmosphere, but the overall rise. And as many who are listening to this will have a geological background, you're fully aware how unique this CO2 level is in the geological history of the last thousands of years. So what you can see here again is CO2 now stored in records in ice in Antarctica over time here in thousands of years. And you can see the waxing and waning of CO2, which is associated with the ice ages. The first time I made this graph was in 2008 when we had reached this point. And today, as you are very aware, we have long passed the 400 ppm. The last time proxy evidence suggests 400 ppm were reached was around 300 million years ago during the Pliocene. And so this was before even Homo habilis was roaming around, which shows the uniqueness of the current CO2 conditions. Climate change is not something for the future. Climate change is already happening. And what you can see here are maps of the impacts of the warming of our oceans over the last decades. What you can see on the left-hand side is the temperature regime, the 1960s to 80s at the top, and compared to the 1980s to noughties at the bottom. And you can see clearly here, if you look at the image of the North Sea, how much the North Sea waters have warmed, because you can see the increase of the red color indicating warmer waters going way into the North Sea. And this has consequences. So what you can see here are records. This is work by the Continuous Plankton Recorder down in Plymouth, one of the most unique data sets of changes of plankton in the ocean. And what you can see here are changes of a warm, temperate, and a temperate copperpot species. And those images are changes through time from here, the 1950s and 80s, to more recent times. And you can see the extent, the distribution of this organism has changed from a species which was predominantly in the Bay of Biscay, going now all the way up to Norway. And similarly, this temperate species has invaded into the North Sea and has gone all along those shorelines. So climate change is not for the future. Climate change is here. Recently, of course, we had a very clear example of how dramatic we are altering, in this case, air quality. What you can see here are two images taken by satellites showing air quality over Europe in response to the recent shutdown we are currently experiencing. On the left, you can see March to April 2019, and the darker the colors, the more air pollution there is. In this case, it's nitric oxide in the atmosphere. In contrast, here in the bottom right, you can see March to April this year, and you can see the dramatic reductions, which shows that humans have direct impacts on air quality in the short term. Immediately, of course, everyone was questioning, how important is this for our CO2? And how important is this? Will we be able to see a positive feedback on the continuous rise of CO2 in the atmosphere? This is a paper which just came out a few weeks ago, led by Corinne Lequer at UEA, where you can see a change in activity here across a number of sectors and regions. So you have power here, but it's a change in activity since the beginning of the lockdowns in those regions. You can see a change in the coal industry, in surface transport, so predominantly cars, as well as aviation, which you can see here worldwide. And most of the sectors, except for the residential, have had a reduction in activity. Now, residential, of course, is increasing because many of us are staying at home. But if you look at the consequences that has, you can see that there are short-term consequences. So here, this is the global change in global daily fossil CO2 emissions, January to the end of April when the paper was submitted. And what you can see here again is the same sector, so power, industry, transport, residential, aviation. And you can see the reductions in the emissions. But if you take this forward, and it's a quite dramatic reduction here. If you look at it summed up for the year, so this is the emissions in billions of tons of CO2 per year globally. You can see these dramatic rises. You can see that forecast for 2020 is a significantly lower emission. And if you compare it to the financial crash, which had an impact, you can see that this is significantly larger. If you're taking it another step forward, though, and you're looking at the CO2 in the atmosphere, not just the emissions, but the reservoir, you will see that there is no indication. This is February, March, April, May. So I took that image from the Mauna Loa Observatory a few days ago. You can see that we still have an overall increase in CO2. So while the emissions are going, some systems respond slower, and atmospheric concentration here are much lower in their response than the air quality was you saw earlier. And of course, again, if we take those values into the long-term comparison, our last decades stand out rather dramatically. It's quite astonishing, given that we have known for a long time that CO2 in the atmosphere is increasing, that it took us until a seminal paper in 1999 to put two and two together and see that if the atmospheric CO2 here in red, again, the Mauna Loa record, is increasing and ocean and atmosphere are in equilibrium with each other, that the seawater CO2 is increasing as well. And if the CO2 in the seawater is increasing, then the pH is dropping, and the pH here in green. It's a very simple relationship, which we are very well aware of, because we understand how CO2 is taken up by the ocean. What we also understand very well is the carbonate chemistry of how how and carbonate mineral is formed. And there's a balance between CO2, bicarbonate, and carbonate ions in the ocean, which is pH dependent. And so the availability of those ions for calcification is changing. So on one hand, we know the ocean is warming, and we're learning the ocean is acidifying. And what I'm trying to show you over the next part of the lectures is examples of what this does to the ocean and what it does to its ecosystems, both today, projected into the future, and in the geological record. Not just because I'm a geologist, but also because I firmly think that there's a lot we can learn from the geological record. It's important to remember that CO2 is not just a negative. Many plants in the ocean, many algae in the ocean are limited with regards to the CO2. So an increase in CO2 is fertilizing. But for calcifying organisms, like coralline algae, coral, and lots of others we are looking at, it changes the energy needed to build the skeletons. And so we wouldn't expect organisms to all respond the same way because they have different constraints. And it's really important that we take this change in CO2 and pH seriously. When you look at the numbers are tiny because we're going from a pH of 8.2, so alkaline, to something which is still alkaline, but less so. So the projections until the end of the century for quite high emission scenarios project a pH drop around 0.35 pH units. This is an exponential scale, so it's really important that you remember that these are large changes. So the changes in protons we've had is around 50%. And then you wonder, does it really matter? Because we as people are exploring every day a really wide range of pH values. So, we touch soap, we might use lemon juice, we have drinking water, and these have really wide ranges. And I'll give you a second to just think about what kind of pH range you think might be covered with this. And of course, this is significantly larger than what you would expect otherwise. So soap is alkaline and has a pH around 10. Drinking water by definition is 7. And then lemon juice is around 2. So we see a huge amount of pH range every day in our normal household items. Our skin is acidic. So we know if we wash it too much with soap, that's not good for the skin. But it's important to remember that many processes in an organism are strongly adapted to one specific value. So a healthy blood level of CO2 results in a pH of 7.4. If that drops to 7.35, you have acidosis and you're about to die. Because every process across the membranes in our body is calibrated against a very narrow pH range. And that's the same is true for many organisms. So what is projected, and is it projected everywhere the same, is one of the questions we need to ask to find out if there will be hotspots where we expect a higher vulnerability. So what we've done here is, again, this is pH, this is a projection for the year 2100. You can see in black on your left-hand side, the global surface pH drop. If you then look at the next one to the right, this gives you saturation. So that's when a mineral is stable, or when you put sugar in your tea when it would stop dissolving, although I don't think we often get to that level. What you can see in blue is calcite saturation, and what you can see in green is aragonite saturation, which is naturally lower than calcite. And they become undersaturated when the value hits 1. And what you can see here are projections for different regions of the ocean, always indicated here in color. So we looked at all regions we considered to be very important to us or potentially very vulnerable. So for example, on your right-hand side, you see the Western Equatorial Pacific, which is the Coral Triangle, one of the biodiversity hotspots of the world. And what you can see here is a drop in both aragonite, which is the mineral corals are built of, as well as calcite saturation, but the values are staying significantly above 1. You could say, therefore, that those organisms might not be impacted because saturation is higher than 1. But again, those organisms have grown and have adapted to conditions which have saturation values significantly higher. Some regions of the world will see undersaturation. So this is the Arctic Ocean, and you can see undersaturation in the second half of the century, as well as in the Southern Ocean. And that is because those areas are naturally low in CO2. So if you think about it, hopefully you all have a similar warm day than we have in Bristol. If you have a water bottle or a soft drink bottle, which is carbonated, you leave it out in the sun, what happens is you open the bottle and the CO2 will degas, so the water will splash in your face. And that is that warm water doesn't hold CO2 or any gases very well. And so saturation is naturally higher in the tropics, while the cold water takes up lots of CO2 and therefore has naturally lower saturation. And therefore we think that those regions of the higher latitudes are very vulnerable with regards to ocean acidification. The other areas which we are very concerned about are areas where we have old water, which is naturally high in CO2, coming up again to the surface, areas which we call upwelling areas. And what you can see here is the Eastern Equatorial Pacific upwelling area. The thickness here of those bars indicates the seasonal variability. So you can see there's a lot of noise in the system, but again, we are seeing significant drops of the CO2. So therefore, if you take all of this together, we see that CO2 results in a warming of the world and therefore a warming of the ocean. That the ocean takes up more of the CO2 and that therefore the ocean will become more acidic. Never an acid, whatever people might want to say. That therefore the carbonate compensation depth, so the depth in the ocean where carbonate is stable, becomes shallower, dissolving carbonates on the way. And other things which are happening is that the stratification of the surface ocean is increasing. So organisms will all live closer to the surface and might therefore be exposed to more light. And at the same time, microbial activity is increasing due to the temperature, and oxygen will get lost in the deeper part of the ocean. So the question then is, what are the biological consequences? There's a vast number of organisms which are important to us, which build their skeletons and shells out of carbonate. And you can see here small microbial organisms, you can see mussels and other organisms here, echinoderms which burrow in the sediments. You can see structural building organisms such as corals and coralline algae. And all of these either use calcite or aragonite. And while the easier part is understanding the chemistry of us burning fossil fuels, which then increases the CO2 and lowers the pH and causes warming, stratification, oxygen loss, our certainty is significantly lower the more complex our questions become. When we're asking about growth, development, reproduction of organisms, and even more uncertain when we're thinking of entire ecosystems and how they might respond, and will things lead to extinction. The other issue we have is that in the ocean, everything really is correlated with temperature. So it's really hard to detect a change. So this is a map here on your top left, which shows net primary productivity anomaly. And you can see a temperature anomaly at the bottom, and you can see the same areas, the upwelling areas, both at the tropical, equatorial ones, as well as the continental ones, in both of those features, because it changed in upwelling warms the ocean as well as decreases productivity. The other problem we have is that we have a lot of natural variability. Depending on where you're listening to me at the moment, it could either be the North Atlantic Oscillation, it could be, some of the, some of the Pacific oscillations, we have lots of change in our climate system on decadal scales. And so when scientists ask for the certainty of when will the conditions be so different that an organism has never seen this, it becomes very hard to state that accurately. And this is what we call the timescale of emergence. This leads therefore to many questions. Will climate change impact biological function? Is one of the climate change factors negative and another one positive and level each other out? Will it affect all the things we are interested in, such as vital rates and health and behavior, in the same way? Organisms aren't the same in every location. What's the role of plasticity? Is it important if climate change happens slowly or fast? Is it important if we have a few days of heat versus many days of heat? And on a longer time, can evolution dampen the impacts? And so all the experiments we do on this will always depend on the context of the location, on the scale. So are we looking at an individual or population? And are therefore much harder to upscale. What I want to show you is some of the results of how we are trying to address this question. And when I say we, I mean we, because this is a team effort of many PhD students and postdocs over the years. What we have done is something which, I do like playing with big instruments, and this is as big as it basically comes. This is a particle accelerator. This is the synchrotron. And so you need a bike to cycle around this here, and you would just see a table down there in the image. And what we're doing is we're taking skeletal material of organisms into the synchrotron, and we are generating CT images, which are comparable of the images generated by you if you had to go in the hospital and get a scan there. What this allows us to do is a really unprecedented insight into the calcite of those organisms, as well as allowing us to generate 3D models and test these with methods which usually would be used by engineers, which are called finite element modeling, which is the underlying techniques of testing the stability of a bridge or if an airplane will fly. And I will show you first a lot of work by Leanne Melbourne, who worked on coralline algae, and then a little bit more on planktic foraminifera, which you can't see, but underneath my cursor, which I hope you can see, is a tiny little foraminifera on this stub in a little bit of foam here. Why we are interested in coralline algae is because that they are extremely important habitat formers on the shelf around the UK. So the dominant species here are Lithotamnium glaciale in the north, Lithotamnium coralloides in the south, and Primatolithum calcareum. These are really important because they build 3D structures where larvae of fish can find refuge, and lots of other organisms. And if you love your scallops, you really have to protect your coralline algae because scallops are usually associated with them. So if we're concerned about warming of the ocean, then what would happen for these species is that they would try to track their habitat distribution and move northwards to stay in the same temperature. But at the same time, when the ocean is warming, it's acidifying, and therefore the saturation will be dropping. And so today, all of the waters around the UK are in waters which are at least twice oversaturated with regards to aragonite. But by the year 2100, we expect this line to be down near Portugal. Coralline algae are important enough that they form where they are marine protected areas. And you can see a huge distribution around them, especially in the north. And so understanding their vulnerability was something we wanted to test here. The first thing Leanne did, and that's really quite supported by the Natural History Museum, was using the collections which had been put in place by the Victorians and having a look at coralline algae grown in Falmouth, in what is now a marine protected area, in the early 20th century, in the mid 20th century, and then went out and dived and got some modern ones. And ultimately what you can see, this is the length of the individual cells within the coralline algae, or the width here, is that there's a lot of plasticity, but not a huge trend. And so that suggests that the organism is able to deal with a wide range of environmental conditions, because of course, the water coming down the fall would have changed. We know from the Western Channel Observatory in Plymouth that the temperatures have changed, but ultimately there isn't a huge change there when it comes to the cell growth. But one of my postdocs then, Federica Ragazzola, is she cultured them, she grew them under conditions which we're expecting to see by the year 2050. And again, we are looking at the length and the width of each of those cells. And the cell walls become projected on up to CO2 of 1,000. You can see the cell wall thickness is falling, and both for within and between the different filaments. Interestingly enough, they still continue to growing even when the aragonite saturation drops below 1. What we also did is we wanted to have a look if the quality of the material is changing. So when you look at here an individual cell, and you then put it into, this is called an electron microprobe, you can map elemental concentration. So again, this is the hollow space where the cell would have been. This is the cell wall around, and you can see beautiful concentric growth. And because of work done by Nick Komenos and others in Glasgow, we know that we can use magnesium in coralline algae to indicate temperature. So the more red this is, the more magnesium, and the warmer the temperature would have been when this is grown. And you can see that within one specimen. When you look here at a summer versus a winter comparison for magnesium calcium ratios, and you can see the warm red colors for high magnesium in the summer part of the specimen, and the blue colors in the winter part of the specimen. What we then also did is we looked at the acidification experiment to see if it would impact how magnesium is taken out. And that's really important because it changes how brittle, how breakable those coralline algae are. And what you can see here are the control conditions with well-defined growth rings. Again, they're overall a little bit lower, but you can see very clearly patterns similar to the summer growth. And these are 580 microatmospheres, which isn't dramatically far away given our current emission. And A, the concentration is lower, and B, this is a very fuzzy image, not because of the image quality, but because the magnesium is much more dispersed. So it suggests that the organism has lost control on its incorporation of the magnesium. So it changes the growth, it changes wall thickness, and it changes the material properties. And what Leanne then did is she built 3D models. So this is basically a series of shoe cartons behind each other, which all symbolize a cell, of the control specimen and of the specimen grown at higher CO2. And she changed the cell sizes to reflect what the experimental data showed. So you have larger cells. That's because more CO2 allows the photosynthesis in this coralline algae to enhance the cells to become larger, and thinner walls. What these images then show is stress distribution. So if it's blue, there's very little stress. If it's red and green, there's more stress. And if it's gray, then there's been a fracture. And what you can see here is an increase in the stress, as you would expect with an organism which has more hollow spaces and thinner walls. What Leanne also did is to compare then the historical material. And so we're back in Cornwall here. This is growth of early 20th century, mid 20th century, and contemporary material. This is an SEM image of the organism so that you can see how it looks like. And what you can see is that ultimately there's no change here in the stress distribution. So that suggests that the function of the coralline algae due to environmental changes has not been impaired yet. She did the same as a comparison for the structure, how those coralline algae, different species, grow up in Scotland. And you can already see here that there's significantly more calcite in those specimens. And that therefore, when you look at the stress distribution pattern, that those organisms in Scotland are much more able to withstand wind or boring or other stresses on their skeleton. So what's important to remember though is not every species reacts the same way. So coralline algae will become more vulnerable, but up to this point they have not. This is a different organism. This is Ophelia pertusa, a deep water coral. Again, grown in the lab. You can see the image here on the left-hand side of the species. You can see the pink where the experiment was started. Again, we took it into the CT scan and scanned it across 3 different pH, 600, 780, nearly 1,000, and there's no change in this organism. And we know that's not the case because we've been able to measure the pH in the calcifying fluid when the skeleton was grown, and the organism is keeping that stable. So there are some species which have internal mechanisms to continue calcifying. And all of this makes it really hard to upscale because we are looking at species which react differently, despite the fact that just purely chemically, we would have expected them to have similar negative responses to ocean acidification. Any of these experiments has one large open question though, and that is what is the potential for adaptation and what's the potential for evolution to help organisms to adapt. So ideally, as a geologist, I would like to go back to the fossil record and have a look at past events of ocean acidification. The issue, of course, is that if we go too far back into the geological record, a Devonian reef doesn't look anything like a modern reef. And so it doesn't have the same structure, it doesn't have the same function. And so we can't go too far back in the geological record. The other thing which was fundamentally important is what we call the mid-Mesozoic plankton revolution. So the advent of calcifying planktic organisms, which help the chemistry of the ocean to stabilize. And so that says that something which is more recent in the geological history is more comparable in the ocean's response to climate change. And then most importantly of all of these is we need intervals which had a rapid change, because if the change is very slow, the organism will adapt to it. Weathering on land will help to buffer the ocean. And yes, there's a change in CO2, but there is no undersaturation. And so this is why often when people question why there's so much carbonate in the Cretaceous is because the CO2 was high over millions of years. And if we take all of these criteria, the event which most of us have studied is the Paleocene-Eocene thermal maximum 56 million years ago. It isn't as quick as the Cretaceous-Paleogene boundary, but as you're all aware, there were lots of other things going on at that time. And so it would be hard to disentangle what's the effect of acidification, what's the effect of warming, and everything else going on. And so I'd like to take you for the last part of the lecture into the geological record of the Paleocene-Eocene thermal maximum. So we went out with the JOIDES Resolution, which is the Ocean Drilling's vessel. She's just leaving Amsterdam, where she's been looked after during lockdown. And we drilled a Paleocene-Eocene boundary. So this is Ellen Thomas. Ellen was one of the people together with Jim Kennett who recognized this unique event. And you don't have to be a geologist to look at the core here and see it's white, it's white, it's white, it's brown, something changed. And something changed quite abruptly. So you can put your finger on this. And this is the Paleocene-Eocene boundary. The beauty of this boundary is that we can trace it on land and in the ocean. We see changes in vegetation, we see changes in the ungulates, it's the base of the primates, and we see a significant extinction amongst benthic foraminifera in the Southern Ocean. Sorry, in the deep ocean, not the Southern Ocean. So what happened? So we think that what happened is driven or is reflected here in this carbon isotope excursion. So what you can see here is, a very rapid excursion, a very rapid negative excursion, which suggests that there was a large input of carbon into the ocean atmosphere system. And if we have a large input of carbon, then this is causing acidification. We also think that it was very rapid on the onset here. And because of that, the carbonate compensation depth changed. dissolved all the carbonate, resulting in these clay-rich layers. We can see that with the onset of this carbon isotope excursion, we have an extinction amongst the benthic foraminifera. This is a very robust group of organisms, so there was no extinction associated with the KPG boundary. We can see, if we look at the images here, this is one species, all the images are at the same scale. You can see a significant reduction in size, so there was dwarfism just after the boundary. And if you look at the oxygen isotope values, they suggest a significant warming. So we have warming and acidification, and we have a biotic response to this event. So it allows us to study in real life the impact of climate change in the planktic and benthic system. What of course is significantly different is the time. So the boundary has moved around. I'm sorry, I have not updated the years here. So this is again the Paleocene-Eocene boundary. This happened over tens of thousands of years. The whole duration of it is more than 100,000 years. So therefore the time scale of impact is different. And so it is not an ideal comparison. This is a figure we've made for the last IPCC report for the ocean chapter. And while the general principles are the same, you have an increase in CO2. This is for the Paleocene-Eocene thermal maximum. This is for our century. We have an increase in CO2 in both of these events. We of course started a much higher baseline during the Paleocene-Eocene thermal maximum. We have a warming. This is here indicated as the anomaly, 6 degrees. Warmings are very significant. And of course, we see warming now in the ocean. But none of our records allows us to have a look at the decadal variability, which I talked about earlier, being very important in modulating this. And so if we're looking at the biotic impacts, they are different. Some of these are the same. So what we can see here is migration of planktic species. This is a coccolithophore record of the Southern Ocean, and suddenly you see warm tropical species appearing in a location where you wouldn't have them before. And this is a record of the last 200 years. This is the Californian borderland, and it's an upwelling area. And again, you can see warm water species suddenly appearing. And so migration of species following the optima is something which was the same in both. But we have very few records of calcification associated with the PETM, mainly because we haven't been able to find, the coral of the PETM as much as we wish. And so we have started to generate some records, and they showed us something rather unexpected, an increase in calcification of this benthic foraminifera. And I'll come back to that example in a minute. Which of course is hard to take as characteristic, given that today we know that species react differently. So this is an example of a coral and the Great Barrier Reef with significantly reduction in calcification. This is an example of a coral from the Caribbean, though, where you see variability which might or might not be modulated by the NAO, which is indicated here in purple. And we have coralline algae, again in the Caribbean, with a modulation there, but not a dramatic loss. So we can't take this example as a one-to-one, most importantly, of course, because we are looking at a significantly longer record. What we know summatively about the PETM is that we had poleward migration of plankton, we had shifts in the assemblage composition, we had novel species appearing, we had migration of mammals on lands, we had dinoflagellate blooms, which today cause red tides in coastal settings. But we had different responses, coastal versus open ocean, and for different species. So what we did is we wanted to have a look at the calcification response, similar as we did it to the modern species. And we took 2 benthic foraminifal species, Natalidis trumpii and Ovidosaurus umbenatus, which live in different, one lives in the sediment, the other one lives on top of the sediment. And we had a look at a couple of its biological characteristics. So its size, its surface area, its volume, its calcite to volume area, the size of the first chamber, which in benthic foraminifera is meant to indicate if we had sexual or asexual reproduction. And then we reconstructed this. And what you can see here is the incredible level of detail of information you can get out of a foraminifera when you reconstruct its development, because its entire development is stored within the shell. And so what we see is that we have smaller species during the PETM, but unexpectedly, these have increased calcification. We took the same approach for a second time interval, 1 million years later, which is half the size of the PETM with significantly smaller input, and we saw no response at all. So the response of benthic foraminifera to calcification, to CO2, depends on the amplitude of the input. So what do we need to do now? And what I think we need to really do is take Earth system models, which we have, which now have ecology in them as well, and start modeling, taking our records, which are core, and this basin versus that basin, and really upscale to the entire world. Ultimately, what we need to do is to be able to go and have a look at the impacts from the physical, where we have such a good understanding, via individual species and locations where we're losing our confidence, to the larger scale impacts for which we need the interaction between the local data records with the modeling to upscale it. Because ultimately what we are tasked with is to go from our understanding of burning fossil fuels, increasing CO2, decreasing pH, impacts on organisms, to the much more complicated to project impacts on socioeconomic systems such as the fishery, the coastal protection, the tourism, and therefore to provide information for UNFCCC and other agencies to assess mitigation options we have. So I hope that I've showed you that there are examples that climate change has already caused impacts, but we've always coped with impacts. So it's a question of how fast and how much. I would like to advocate for decision-making despite uncertainty about when things are happening and how bad things will be happening, because the longer we wait, the more difficult it will be to deal with it. I think the geological record shows very clearly from our example that many ecosystems have responded with geographic changes, seasonal changes in seasonality, migration to climate change in the past. And despite the fact that the climate change was slower, therefore suggesting that our current anthropogenic climate change will have significant impacts on ecosystems and species. And I hope that lots of geologists will produce papers which will support the next IPCC assessment. Thank you very much.
Hey, thank you so much for that talk. We've had one question come in so far, so I think I'll read that out. And if anyone else has questions, please do submit them in the Q&A box. So our first question is, are planktic or benthic organisms more at risk of ocean acidification today with an atmospheric input of CO2? For example, why are benthic organisms affected during the PETM, but are largely unaffected in the KPG event?
Yeah, that's not a simple question. we think, so A, we could see that species which live in the sediments might be less impacted, mainly because we think that they have already dealt with low saturation conditions all the time. But why the PETM was so much more important, we think, is a combination that also because the ocean, the bottom ocean warmed rather dramatically. And every organism needs, doubles its metabolic rates every 10 degrees. So therefore it needs twice as much food every 10 degrees. And if at the same time the ocean stratifies and doesn't provide that food, then this is not necessarily a question of acidification, a little bit like the example of the deep water coral I showed. As long as you feed things well enough, they can deal with lots of stress. And so it's the combination of many factors coming together which leads to the extinction of the benthos during the PETM. Which one is more affected is really difficult to say, because you could make the argument that the acidification will be much more rapid and strong in the surface ocean and then propagate slowly to the deep ocean. On the other hand, surface organisms are used to changing climates. They see diurnal changes, seasonal changes. Benthic organisms have a very stable environment for their entire life cycle.
Great. We have one from the chat, which is how well can we assess the uncertainty produced in modeling of the climate ocean system due to not being able to represent well or have a limited understanding of the impacts on the ecosystem and the potential impacts that these ecosystem impacts could have back into the whole system. If we need to encourage policy decisions while there is still uncertainty, then how well we know the uncertainty will matter hugely.
That's again a very good question, because if CO2 changes photosynthesis, then it will change primary productivity. And therefore will change the entire food web. It will also change what we call export production. So the part of the primary productivity which is exported to the deep ocean and locked away there. Then some people have suggested that for organic matter to be exported to the deep ocean, we need a ballast. So we need a shell. And if that shell is thinner, or test in case of a foraminifera, will it still allow it to sink? This is very much at the forefront of where those combined ecosystem, ocean ecosystem models are currently going. Up to this point, many of those models would have had one characteristic diatom and one characteristic coccolithophore in them to model the shell-forming plankton. But we know that all of these species are responding differently. And so therefore, this is not a sufficient characterization, and therefore the uncertainty is high. The risk is that we get it wrong. So every time you breathe, every second breath is coming from the ocean. If we get this wrong and we're impacting ocean ecosystems, we're impacting the entire world.
I have another question. Do you think that the organisms that seem not to be affected by ocean acidification use more energy to maintain their shells, and that there may be limits/tipping points of pH at which they may not be able to maintain their shells?
Yes, the answer to that, we have seen in experiments in the labs that if you increase feeding, there's, for example, really no effect of high CO2 on bivalves. But all of these are usually done for a few weeks. And so it's absolutely true that at one point there might be a tipping point. More importantly, though, in the ocean, and the higher CO2 goes hand in hand with increased stratification. And increased stratification reduces how nutrients are brought back to the surface of the ocean, and therefore the productivity. And so there will be less food. And so while this is lovely to do as an experiment in the lab, it doesn't reflect the reality of food limitation while energy demands are increasing.
Great. Oh, questions are coming in. Are you more worried about the impacts of ongoing climate change/ocean acidification on open marine plankton or on coastal coral reefs, which is more vulnerable?
You're basically asking me to choose between my two children. So the question is, are we more concerned about ocean biogeochemistry, or are we more concerned about loss of biodiversity, loss of coastal protection, and fishing in areas where a huge part of the protein those people have is coming from the fish which is associated with the coral reef? So there is no answer to it. I think it's really important that we think about the ocean as one big space we need to preserve. And there's an initiative currently going on which is called 30 by 30, so 30% of the ocean under protection by the year 2030. Because what we understand is that you can't just have small parts protected, because those organisms will migrate. They will go, they will follow their habitat as much as they can. So if the next part isn't protected, it's not going to work. And in the UK, our marine protected areas depend on the species. So they're protected for the species. They're not, if that species isn't there, although this will be the right habitat in 10 years' time, you can't protect it. And so we really need to think differently. We need to think about this as an interconnected system. So you can't just pick one over the other. And we need to think about making protected areas and making sanctuaries for tomorrow, because the organism will not just stay static.
Great. And another one, so did oceans and marine organisms experience other similar conditions in the past for elevated CO2 and temperature? And do those past events have any reflection on the current research on the topic?
So there have been time intervals where the CO2 was much higher than what we even projecting for the end of the century. But as I said, it's ultimately not about the absolute value, it is about the rate of change. If the rate of change happens quickly, then the pH in the ocean and the saturation of the ocean decouple and go different ways. And that's when it becomes really difficult for the organisms. So the Cretaceous, for example, the most famous Cretaceous rocks in this country are the White Cliffs of Dover, were deposited under very high CO2, but those organisms have been adapted to that CO2 for millions of years. So I'm not concerned about the CO2. I'm very concerned about the rate with which we get to that CO2. What we've also learned in extinctions, it's always the most specialized species which goes first. The generalist does much better. Humans are an incredibly specialized species.
Great. And then if we are able to reverse increasing levels of CO2, is it better to do it quickly or slowly?
I'm not completely sure I get the question. So if it's about reducing our emissions, then ideally as quickly as we can. Because the time, a lot of people are, for example, talking about planting trees to draw down CO2. A tree doesn't do that for the first years. And so any kind of method we are coming up with takes time. There are some experimental sites where people are putting CO2 into oil fields. Of course, the oil companies are interested in it because it increases the pressure on the oil field and therefore gets more oil and gas out. That's called carbon capture and storage. Other options for carbon capture and storage are in saline aquifers. All of this is usually tiny little test sites. And whatever we decide in the risk balance for mitigation, we need to think about the fact that currently we just have tiny little test sites, and this needs to be a much bigger, much more global effort to ensure that it reaches the amounts we need. I mean, a global lockdown had an impact on the CO2 emissions, but it just brought us back to the 80s. We need to go back much further.
And I think I'll do one last question, which is, you said that there is a migration of 4M species at the PETM. Are we seeing this happen under current climate change? Do you think that with the rate of our current climate change, we will see extinctions rather than migration?
So yes, we see changes, not just amongst forms, amongst many organisms moving northwards. And we also see this significantly faster on land. And that might be because marine organisms just can use ocean currents, while land organisms have to get over mountains and roads. So we definitely see that at a very fast rate. What we haven't seen yet is a climate change linked extinction in the ocean. Of course, there have been many, but they're predominantly overexploitation. And so am I concerned that we will have climate change caused extinctions? Oh, yes. But we haven't had them yet. So it's in our hand to prevent it.
Great. I think looking at the time, we'll have to draw this to a close, but I want to thank everyone for having such a great conversation. And of course, thank Daniela for such a fantastic talk. We've really enjoyed having you all here, and we hope to see you at our next virtual event. So thanks so much. And thank you, Daniela.
Okay.